let's do a quick revision on post-transcriptional modification. What basically the post-transcriptional modification is and the steps. We know in eukaryotes from the DNA to mRNA is produced through the transcription process and it occurs on the nucleus. Okay, so the first stage of the mRNA, we call it the heterogeneous mRNA. Sometimes it is known as the primary transcript. This heterogeneous mRNA basically non-functional. To become functional one, it undergoes some modification. The first modification basically called the 5' capping. This mRNA has 5' end and the 3' end. On the 5' end there are modified guanine nucleotide basically added, the 7-methyl guanosine. That nucleotide added on the 5' end. Sometimes the 3-phosphate group also added, triphosphate basically, and it prevent from being degradation of mRNA. Okay, and then the 3' polyetyl, we called it polyadenylation. Here on the 3' end of mRNA there are basically 200 to 300 adenine nucleotide on the string of 200 to 300 adenine nucleotide basically added one by one, and it helps and provide stability. So the mRNA basically gets stability whenever the 3' polyetyl is added. Whenever these two modifications occur, like 5' capping and 3' polyadenylation, these heterogeneous mRNA become pre-mRNA. Here the introns are still present. Okay, so there need one more modification. The non-coding part we need to remove. The introns are basically the non-coding part. We need to remove these introns, so the splicing occur. Splicing means removing of introns and joining of exons together. So whenever the splicing occur by the spliceosome complex, here basically small nuclear ribonucleoprotein snRNPs basically work. Some proteins here work, basically some other proteins also. Whenever the splicings occur, then this pre-mRNA become mature mRNA and it is ready to leave the nucleus. It binds to the ribosome to translate the protein. Okay.